I'm going to start on this slide, which is taxonomy. So taxonomy, Linnaeus was the scientist that developed the way that we organize and name, which is called taxonomy, so the naming of organisms. So this was around the time when people were just going out and they were discovering things. So he figured we needed a way to organize organisms that people were finding, like does it have fur, does it have live babies, does it have eggs, or how to organize everything. So Linnaeus is the one that came up with that and developed it. So in micro, most of the time what we're going to call organisms is we're going to call things by genus and species. So if you think about Staphylococcus aureus. Okay, this is the genus. Okay, and then the second part is the species. So it would be the same thing for, say, Escherichia coli, which is E. coli. Okay, so Escherichia coli, the genus is the first part, and the species is the second part. Okay, if you're typing something, it's always going to be, the genus and the species should always be italicized. Okay, if you're handwriting it, it needs to be underlined, okay, so that you know it's the Latin. The first word, the genus, the first letter is always capitalized, and then the second word, the species, this is lowercase. So the E is capitalized and the C isn't. The first time you write something out, you always type out the whole thing or write out the whole word, and then the second time after that, then you can use like E. coli. Okay, and that would still be underlined or italicized. Okay, now viruses. So viruses aren't considered living. So viruses have their own naming system. It's totally different. There's a worldwide viral naming group. Lots of times viruses are named after the symptoms that they cause. So one well-known virus is called tobacco mosaic virus, which is also known as TMV. So it's a plant disease, and what it does is it affects the leaves of the tobacco, so they look kind of like a mosaic. They don't look totally green. So we have a whole chapter on viruses, so we'll talk more about how they're named and how they're named and why they're named that way. Okay, now this is a list. She goes over all of this in the textbook. So functional microbiology. So these are different fields of study that might use microbiology. So the first one is immunology. So you guys know what immunology is. So immunology is like the immune system, how it works. Epidemiology is the spread. Okay, so epidemiology is how diseases spread. And there's a whole chapter on it. How diseases spread. Etiology is how they're identified, how a certain disease is identified. Like right now, Zika, right? So how are they determining that this virus that they're calling Zika is what's causing these symptoms? Okay, bioremediation is using microorganisms to fix something in the environment. So an example of that would be a wastewater treatment plant. So the wastewater is remediated or kind of fixed or taken back to where it needs to be using microorganisms. Okay, every hospital, tattoo shop, dental office has someone that deals with infection control. So it's someone that understands how microorganisms spread, what it takes to grow them, how you can clean things up. So they're kind of like quality control. Chemotherapy. So chemotherapy, you guys are familiar with using drugs to treat cancers. And initially started out as using drugs to treat any disease, so using chemicals. Industrial microbiology can be where maybe they're growing an organism to make yogurt, or they're growing an organism to get enzymes to put into detergents. There's lots of different kinds of industrial microbiology. So biotech we'll talk about, so that's using living organisms to, usually it's to help somebody. Agriculture, so agriculture it could be causing plant disease, it could be causing animal disease. There's just a ton of different things that are out there. Okay, now the history of micro. So it's really complex, and it's been going on forever. So a lot of diseases have determined what happens in the future. So like Black Plagues, bubonic plague, they've all determined like a whole population can be wiped out or decimated. And that's going to determine like where do the people go, what happens next. So that's just kind of like, in general, microorganisms have had a lot of effect on like people and society. Okay, now, two important men are Robert Hooke and Anton von Leeuwenhoek. So these are the two people that are given credit for developing microscopes. So Robert Hooke was the first person, like they were lens grinders. So they learned how to grind glass to make a lens. So that was the important concept that they needed to get, was how do you make a lens, and how do you grind the glass so that you change the shape so that it will magnify. So Hooke, he is credited with coming up with the term cells. So he looked at cork under the microscope, and he could see these little squares. Okay, so these little squares, he thought they looked like the rooms that the monks lived in the monastery, which they called cells. So that's where the term cell came from. So he made the simple microscope. Now, Anton von Leeuwenhoek, he was a Dutch merchant. So he sold stuff, right? He just kind of was a trader, like a businessman. But on the side, his hobby was grinding lenses. And he figured out that if you put more than one lens together, you could increase the magnification, like you could double the or multiply the magnification. So he is credited with the first compound microscope. He must have been an interesting person. Like, he didn't share a lot of his experiences, so he kind of like kept what he did to himself. So it took a while for the compound microscope to get out and for more people to be able to use it. It's cool. There's a museum in Bethesda. It's called it's a military museum. I can't remember exactly what it's called, but they have a huge microscope collection, so like the biggest one in the world, and they have some of von Leeuwenhoek's original microscopes there. Okay, so early microbiologists, so Linnaeus, we've kind of talked about, and that he did taxonomy, and then Schleiden and Schwann were the first people that they came up with the idea of cell theory. Schleiden worked with plants, and Schwann worked with animals. Okay, so they were coming up with the concept that you have to in order to get a cell, you have to have another cell. Like, cells just don't come out of the air. That you have to have one, and then it's going to divide, and that's how you're going to get two. They also came up with the idea that like cells are the smallest unit of life, and that you can put groups of cells together to make an organism. So these guys are the ones that came up with the idea of cell theory. So one was working with plants in one place, one was working with animals, and it turned out that, they were coming up with the same ideas. Okay, now, spontaneous generation. So back in the day, before they understood science and that the scientific method existed, people believed that if something bad happened, if someone got disease, it was because they were bad and God was punishing them. So scientists didn't necessarily believe that, but they needed to prove it. They needed to demonstrate that cells come from other cells or organisms come from other organisms. So Redi was an Italian, and he had this classic experiment with meat. So he was trying to disprove spontaneous generation of maggots. So people would think, okay, if you did something bad in your house, your meat would get maggots, and then you would get flies, and your meat would get destroyed. So to disprove that, he just had 3 jars with meat. Okay, one was open, so the flies could come in and they could lay their eggs. Another one was corked, so the flies couldn't get in, and there were no maggots. And then there was, the 3rd jar was covered with cheesecloth, okay, so like gauze. And so the flies were still attracted to the meat, but they laid their eggs on the surface of the cheesecloth. So he's like, oh, look, you have meat, but you don't, the eggs are not coming out of the meat. The flies are where it's coming from. So that was, it seems really, really simple, but that's one way. Pasteur also worked on this idea. So Pasteur, what was the idea? He had a flask that was specifically blown, like the glass was blown like this. It's called a swan neck flask, so it's got this real long neck. And the idea was they could put sterile like chicken broth in there, and air could get in. Okay, so the air could get in, but the organisms couldn't get through. So what happened is he just kind of proved that he could keep the organisms out and maintain sterility, so that it was the organisms that were causing the problem. Okay, Pasteur is also really well known. He's the one that figured out that the good yeast, the good things growing on the grapes, make good wine. He's took care of pasteurization, came up with the idea of that, which we'll talk about more, and he also developed the rabies vaccine. Okay, so germ theory is the idea that germs are going to cause disease. Bacteria or living things can cause disease. It's not that you're bad and you're being struck down by God. Koch's postulates. Koch came up with the idea of these rules in order to prove germ theory. So Koch says a specific microbe can always be found associated with this particular disease. Okay, so you're always going to be able to get that microbe back out of a diseased organism. Okay, the microbe can be isolated and it can be grown in a lab. And then if you take what's growing, that pure culture out, and you put it into a susceptible animal, okay, you should be able to get the disease back. And then you should be able to recover the organism again. We don't have to do this as much anymore, right? Because most of the diseases that we know, the etiology, everything's been figured out for them. So it's not too common that we have to go through and do Koch's postulates. It's hard to do with animals right now, right? So animals and humans, could you just do this with a human? Oh, yeah, you're healthy. Let's see if you get the same diseases so-and-so has, because we're going to put the bacteria in you. We don't do that, right? It's easier with plants, or insects, but we don't do it with humans very often. Okay, now, Semmelweis and Lister, these guys came up with the idea of aseptic technique, and they helped come up with the idea of hand washing. Okay. Semmelweis is the first one. So he looked at what's called childbed fever. And childbed fever, what happened, so this was in like the 1800s, and in France, and I think that's France, what happened was there were, usually for a while, women had the babies at home, and the midwife would come and help with the birth, or the family would help with the birth, and things would be pretty okay. So then hospitals were developed, and it became like cool, or I don't know, kind of like explained how affluent you were, or like how much money you had, if you could go to the hospital to have the baby. Well, it turns out that the moms that had their babies in the hospital would get an infection and a fever and would die more frequently than the people that were being delivered by midwives. So Semmelweis noticed this, And he started to try to figure out what was going on. So he noticed that the surgeons or the doctors were not washing their hands or like taking off their coat or their apron between patients. So in some cases, they were even doing autopsies with dead bodies and then just running up and delivering a baby with their dirty hands. So they were introducing bacteria to the mom when they were delivering the baby. So Semmelweis, demonstrated this and said, you need to wash your hands. You need to, there are things we can do to prevent this. But it was not widely accepted. So he was the one that was doing it, but people didn't want to believe him that that's really what was happening. So he ended up actually he lost his mind and he ended up in an institution. And I think he ended up dying of an infection. Okay, then Lister came along a little bit later after Semmelweis had floated those new ideas, and Lister came in and was like, yeah, we need to wash hands. We need to make sure that there's a clean field when we operate. And by that point in time, more people were accepting of the ideas. So Lister's he's the aseptic technique, hand washing. He gets the credit for it. Okay, there are all sorts of future trends out there. So we use recombinant microbes to make drugs, to make hormones, to make vaccines. And recombinant just means it's two or more organisms put together. Or sometimes like the same two DNA from two sources put together. Okay, we talk about gene therapy. We'll talk about a little bit. Bacteriophages. So bacteriophages are viruses that infect bacteria. We'll talk about more. And then genomics. And genomics is studying the whole genome of an organism, so all of their DNA. So there's a whole chapter on this that we'll hit on. But there's there's lots of things that are changing. It's not a static field. There's lots of stuff happening.